Welcome to GeOME demo number 1, where I'll show you how to upload metadata that describe either biological samples or sequence data generated from biological samples. So here we start off on the home page, and the first thing we'll want to do is to create a new account. The next thing will be to define a project. A project can pretty much be anything. It can be all of your lifetime output as a researcher that you want to submit to GeOME. It can be all the output from a particular grant. It can be all of your consortium members. If you have a group of collaborators that work together on using the same standards, they can all work in the same project. All those things would make a good project definition. And for now, I'm going to keep this project private. And now I have to choose a configuration. A configuration basically tells us how you want the data to go into the database and how you want it to come out of the database, as well as some validation rules. So we have several choices here. We have two generic choices, multi-sheet and single-sheet generic. And these have a minimum number of required fields and are basically customizable to whatever set of standards that you want to use. The difference between multi-sheet and single-sheet is simply whether you want all of your metadata to be out on one single sheet, spreadsheet style, or whether you want your metadata to be related to each other via primary and foreign keys in sort of a relational database style. In multi-sheet generic, there will be sheets for each sample, each sampling event, and each tissue, whereas in the single sheet, that's all kept on a single page. There are also three configurations that are kind of presettings that have been made for various purposes. The individual-based sequencing and barcoding is a single sheet. Population genomics is also a single sheet. And biocode is a multi-sheet configuration that was designed for eDNA and metabarcoding. So I'm going to choose the population genomics single sheet configuration, and I'm going to keep my configuration in sync. This means that any changes that happen to the configuration will be propagated into my project. If I keep it out of sync, then I can customize my project to my particular desires. So I will create this project now. And if we go down to the project overview, we don't see much there yet because we haven't added any data. If we go to the project settings, we can see the name, whether it's private or public. I can choose to take it public anytime that I want. There's no expeditions yet, and there's just myself as a member, although I'm allowed to add other members to the project. So the next thing we want to do is to generate a template. And a template is basically, at the beginning of your project, it can sort of serve as a data management plan that you know ahead of time all of your collaborators will also be using, and therefore will make your metadata compatible not only with your collaborators, but with everyone else who uses GeOME. And we have a wide variety of possible fields that can go into your data template. These fields are taken from Darwin Core, which is a body of standards that is intended to facilitate the sharing of information about biological diversity. And these allow you to be sure that your metadata will be compatible with data that are collected by other labs. So you can see that there are some pre-checked fields. Those are the fields that we require. There are also some fields that are uncheckable but are already checked. We recommend those fields. For example, I don't have any information about the sex of my reef fish, so I'm going to uncheck that one. And you can see that there's actually a definition given for every one of these fields to be as clear as we can about what we mean about each of these fields. If you come down here, you can see that we recommend that you give us dates. I actually don't know month and day that these were collected, so I'll uncheck those. We require either a locality or a decimal latitude and decimal longitude. For example, you could also include habitat. And if you look at habitat, we've defined habitat following the environmental ontology with a wide range of possible habitats that your samples could have come from. Okay, so I'm happy with all of that, and now I'm going to come back up here and export my template. And when I open it up, I first see an instructions page that tells me about the various tabs in my spreadsheet. Then I have a samples tab. In this case, I've chosen a single sheet sort of setup, so all of my metadata are on a single sheet. The red fields are required fields, and the black fields are not required fields. And this is where I'll actually fill in my data. In the samples field, there are all of the fields are listed together with their definition and their controlled vocabularies. And if we come over to lists, we can see all the controlled vocabularies associated with various fields. All right, and now cooking show style. I actually have some data kindly lent to me by Dr. Michelle Gaither. And we can look at a completed metadata sheet where I have now filled in phylum, principal investigator, year collected, locality, as well as latitudes and longitudes for all of these samples, all of the other information. Notice that there is some missing information for life stage. That's okay, that's part of research. And all of these material sample IDs are locally unique to this particular set of metadata. So each one of these should be a unique identifier. And if I go look at the other file here, I've also created a file that is a list of all of my material sample IDs as named FASTQ files. So this is a list of FASTQ file names where each one of these file names corresponds to one of the individuals on my material sample data sheet, a data column. And GeOME will validate that each of these is, in fact, does have the same name and that they're all present. So I've got my data ready, and I can go back here and go over to load data. And I have an Excel workbook that I want to load. And I have FASTQ file names that I want to load. And now I'll pick my file out. And as soon as I load my metadata file, you see it pops up a map that shows where the lat longs occur on the globe. And this is a great chance for me to check each of my lat longs to make sure that I've entered them correctly. And I can zoom in and look at particular lat longs and which individuals are included there. And this would be the place where I'd be alerted if I had some problem with my geographical information. But for now, these all look good to me. I'll then browse to my list of file names, import those. This is a single-end RADseq library. And we will choose WGS strategy, genomic source, reduced representation, RAD library. This is an Illumina, MySeq, and I'll add the citation to the protocol. And then I need to create an expedition for this. So an expedition is sort of a sub-project. And I think of expeditions as either all of the samples that you get from a particular sampling expedition, or possibly all of the samples that were used in a particular paper. So I'll define this by paper. and just name it after the paper that was published. So we will create that expedition. And now I'm ready to load my data up into GeOME. So you'll notice that it does some validation, and it's warning me that there was a missing life stage. We noticed earlier that there were some missing cell values there. That's okay. It's just giving me a warning. And also, I removed sex from the sheet. That's fine. I also don't know month or day collected. I knew all that ahead of time. I'm glad it told me, but I can continue. And now it's prepared some files. It's uploaded the metadata, and it will propagate those metadata all the way into GBIF and keep them as well in GeOME. And then I can download these GenBank files. And when I open these up, I see I have tab-separated data. The two attributes and metadata files that are required by GenBank, and they're all formatted and ready to be uploaded. And there's a set of instructions as well that will allow me to move over to GenBank and upload these data into GenBank. So now I have created my user account. I have created a project, selected a configuration, generated a data template for that configuration, filled out the data, validated it, uploaded it, and now have gotten a GenBank package ready for submission to the SRA. Once the data are submitted to the SRA, they will be GeOME will go and harvest the accession numbers and create a permanent link between the metadata uploaded here and the sequences on the SRA. So thank you for your attention. The next demo will be on how to download and search and download data from GeOME.